I got it. Okay.
I do not see the, I see the waiting room now. Okay,
Could have popped up. Yeah, okay.
thanks.
Okay. We'll go ahead and start. My name is Jess Sappington. I'm the Food Systems Program Coordinator for the Regional Small Farms Program, housed out of WSU Extension. Tonight's our latest online Dirt Talk event, where we'll be diving into the topic of slugs and snails. For those that have not attended a Dirt Talk previously with us, events are hosted by our WSU Regional Small Farms Program in conjunction with a local farmer or ag specialist who's willing to share their expertise on a given topic. Tonight, we're super excited because we're joined by Oregon State University Associate Professor and Extension Gastropod Specialist, Rory Mcdonnell, who will be sharing his knowledge and research on slugs and snails and how to control them on your farm. We also have WSU Regional Small Farms IPM Specialist Laurel Moulton here with us, whom some of you have already met, who will be on the call. She'll be taking part in the discussion and the question and answer session at the end. So feel free to utilize the chat box throughout this whole discussion, and we'll make sure to stop at points throughout to answer questions about that come about from the group. But we will definitely have plenty of time at the end for a discussion. The events are meant to be interactive, so please don't hold back your questions and take your chance to put it in the chat if you have the opportunity. Okay, we're going to go ahead and we're going to get started with Rory. Rory, do you want to go ahead and introduce yourself and start us off?
Will do, Jess. Let me just make sure I'm sharing the right screen. Share. Is that looking good? Awesome. Hello, everybody. My name is Rory Mcdonnell, as Jess said. I'm an associate professor in the Department of Crop and Soil Science at Oregon State University. I've been working on slugs and snails for the past 15 years. I've worked on these organisms in Europe. I'm originally from Ireland. Also in Southeast Asia, South America, and in the United States as well. So I do want to say thanks to Jess for inviting me to come along and to speak with you all today. It's exciting to be able to talk to growers in other regions outside of Oregon. So I'm excited to learn about your issues and your concerns and the issues you're having with slugs and snails. So over the next 45 or so minutes, I'm going to introduce you all to the weird, wonderful, and sometimes downright bizarre world of slugs and snails. Going to start out by giving you some background information on these invertebrates, and then we'll take a look at some of the basics of slug and snail identification. Also want to talk to you about some of the most damaging species we've got in the Pacific Northwest and show you how you can identify them. Going to move on then and talk to you about current control options and give you some tips and some advice. And to finish up, I want to tell you about some of the novel approaches that my research team and I are currently developing to help us manage these pests in agricultural systems. I like to have interactive presentations, and I do have a number of questions scattered at various points throughout my talk. And when we get to the first block of questions, Jess will explain to you how you answer the question. During this presentation, I will be mentioning the word gastropods quite a lot. And when I say gastropods, essentially means slugs and snails. Okay, so over the course of human history, we as a species have had a very interesting and sometimes even intimate relationship with slugs and snails. Over the centuries, we've used them in rituals, primarily in the South Pacific, which I believe also as a skin treatment, primarily in Southeast Asia. And slugs and snails make appearances in lots and lots of our medieval art. My wife, she's from Spain, and in that part of the world, people like to eat snails. They're considered a delicacy called escargot. And then in other regions, in places like East Africa, people grow giant snails in really, really large numbers because they're a very important source of food and very high in protein. So in our society, slugs and snails have played, and they're going to continue to play, a very important role. A question that I'm always asked when I give talks like this is, Rory, how did you get interested in working on such disgusting creatures as gastropods? Well, I first became interested in slugs and snails when I was a young lad growing up back in Ireland. I had a very, very good collection of natural history books, largely thanks to my dad. And I can still to this day remember reading in one of those books that slugs and snails can crawl easily over broken glass, and they have teeth sharper than those of a shark. So when you're young, you're already interested in natural history. These sorts of facts tend to get you completely hooked. And I guess I've never really looked back. Today, their repulsive nature has made them something of a celebrity, and there have been many Hollywood movies and even some books which have had slugs or slug-like creatures as their central characters. Now, slugs, believe it or not, have also given rise to a number of phobias, and a quick check in the American Journal of Clinical Hypnosis shows that there have been many cases where prolonged hypnotherapy is needed by patients to overcome their fear of these organisms. So you and I, we don't like slugs and snails because they feed on plants in our garden, they feed on our crops, but some people have a real pathological fear of these organisms, and they need to seek medical treatment in order to get over that fear. So I'm guessing if I was to ask you all, what is a slug? And most of you would tell me it's essentially a snail minus its external shell. And you'd be absolutely right. But from an evolutionary perspective, this might seem like a bad move. And surely losing a protective shell is not good for an organism that's comprised almost exclusively of water. But when you think about it, it does have a number of very distinct advantages. So for example, slugs can crawl through remarkably tight spaces. And because they don't have the same calcium requirements as snails, they can also live in a much wider range of environments. And because they're not carrying a heavy shell around in their back, they can also move a lot quicker than snails. And that can be particularly useful if you're trying to escape from a predatory invertebrate, for example. I know you all know what a slug looks like, but I do want to highlight the main body parts because some of them are important when it comes to identifying different species of slugs. So we know the vast majority of slugs do not have an external shell, but many people are surprised when they learn that slugs actually have an internal shell. The vast majority of slugs actually have an internal shell, and it's located underneath this saddle-shaped structure at the front end of the slug that we call the mantle. The mantle also has a breathing pore. It's sometimes called a pneumostome, and that's almost always located on the right-hand side of the slug. At the front end, there's two pairs of tentacles. You've all probably seen them. And the upper pair, we call these the ocular tentacles, because the slug uses them to detect light. But we also think that slugs use these larger tentacles to detect odors in the air. So they act a little bit like a nose. Lower tentacles, these are called the sensory tentacles or the peduncular tentacles, and the slug uses these to taste. So they act a little bit like a tongue. Other important features include a keel, and that's a raised ridge that can run from the back of the mantle all the way to the tip of the tail. At the tip of the tail, we sometimes have what's called a caudal mucus pore, and that's a very large pore that's typically filled with a thick mucus plug. The tubercles, and these to me look like the building bricks of the body. And then just in case things aren't strange enough with these animals already, when they're getting ready to mate, their genitalia literally fall out of the sides of their heads through this pore. It's called a genital pore. Now, the vast majority of slugs are hermaphrodites, so they have both male and female reproductive organs, so they can self-fertilize. The structure of the genitalia is really, really important when it comes to identifying closely related species of slugs. I'm not going to get into that level of detail with you today. It's quite technical, and there's no real need to do it today. In terms of snails, again, I'm sure you all know what they look like, and they do have most of the same body parts as slugs. So, for example, snails do have a pneumostome. You can see it underneath the shell, and they also have a mantle. Again, it's located underneath the shell. They also have upper ocular tentacles and lower sensory tentacles as well. But the most striking difference with slugs is that snails have a very obvious external shell, and that shell can take on a variety of different forms. It can be long, thin, and cone-like. It can even be globular, and in some species like amber snails, it can even be turret-like. Right. One of the things that I've been doing since I first got to the US about 10 years ago is I've been trying to get a handle on what species of slugs and snails are causing the most damage here on the West Coast. And not surprisingly, it's the invasive slugs and snails, and the vast majority of these are accidental introductions from Europe. And most of them accidentally hitchhiked here, many unimported shipments of plants, others, would you believe, unimported shipments of tiles from the Mediterranean region, and others still on equipment that's being brought back to the United States from military bases in places like France, Spain, Germany, and Portugal. I don't have time, obviously, to go through each and every single pest, slug, and snail species on the West Coast with you this evening. So instead, what I want to do is I want to talk to you about some of the more important ones, some of the more interesting ones, and show you how you can identify them. Before we do that, I do have some questions for you folks, just to get an idea of your opinions on slugs and snails and your knowledge of different species.
Perfect. So for those of you who have not done polls before, I'm just gonna put up a question, a poll, and then you're gonna have options to answer. And then we'll give it a few minutes, not a few minutes, probably about 30 seconds actually for you to input your answer. And then we'll go ahead and share the results in real time. So I'm gonna go ahead and launch question 1. Can you all see that? So question 1 is, considering overall costs and damage, how would you rank slugs and snails as a pest on your farm? Go ahead and click your answer. And as you can see, getting some real time. Rory, can you see the real time answers coming in?
I can't, Jess, I can just see it says 0 of 1 answered, so I can't see just yet. Maybe I'll answer as well, see if that helps. So I don't see anything just yet.
Okay, I'm gonna go ahead and end the poll. It looks like we've got 93% of the participants who've clicked that button. So gonna go ahead and end it and share the results.
Excellent, okay.
Okay.
Thank you, folks. So 37% of you said that slugs and snails are the worst pest on your farm, 22% second worst, and 15% say they're the third worst. So these organisms are obviously causing you guys a lot of issues. Okay, that's really good for me to know. Thank you.
Perfect. Okay, we're going to go to question 2, unless you wanted to talk more about that.
No, that's perfect, Jess. Thanks.
Okay, we're going to go ahead and launch this. You should be able to see question 2, which are most damaging on your farm. Please select from slug snails, both slugs and snails, or slug snails are not a problem for you on your farm. Looks like we got some people still answering. We'll give it another few seconds. Okay, we'll go ahead and end the poll and share that.
Excellent. So this is typically the response that we get, particularly when we use this question in the Pacific Northwest. Slugs tend to be a lot more problematic than snails. And snails can sometimes be pests, but in field crops and so on, slugs tend to do the most damage. Interesting that nobody says that slugs and snails are not a problem on their farm. Okay, thank you.
Okay, so we're going to go to question 3 now. And this question is identification. So how confident are you that you can correctly identify different slugs and snails on your farm? Please go ahead and select your answer. Still a few folks clicking. Okay, I'm going to go ahead and end that poll. It looks like we've got a good portion. We're right about the 93% of participants given answers here. I'm going to share that.
Oh, very good. So most of you know 1 or 2 species. That's impressive. 8% of you know greater than 5 species. That's really impressive. So what we're going to do now, the next couple of questions, I'm going to actually test your knowledge and to see if you know 2 of the most damaging slug and snail species there on the West Coast.
Okay, so we're going to go ahead and launch this one. So if you can't see the small version of this, Rory has a larger version on his slide. So go ahead and move your poll window if you're not able to see. Let's go ahead and select your answer there.
And there are 4 different choices for this particular specimen. On my screen, it's just showing 2 choices, but if you scroll down, you can see the other 2 choices.
Right about. Give it another second here if anybody wants to chime in on theirs. And we'll go ahead and end it. We're at about the same percentage of people taking part. I'm going to share that.
Very interesting. So this slug is actually the grey field slug. 28% of you got it correct. Well done. 44% thought it was a white soul slug, which does look a little bit similar to the grey field slug. So 28% of you got it right. The grey field slug is one of the most damaging species on the West Coast, and it is a species I'm going to be talking to you about later in my presentation. So we'll have more on that in a few slides' time.
Perfect. So I'm going to go ahead and do this next one. And this is asking you, what is this snail? There should also be 4 options for you to select from on this one. Let's see. Having less people answering quickly on this one, so I'm wondering if we're thinking. Looks like the answers slowed down, so I'll go ahead and share that poll with you guys. Okay.
Wow, very impressive. So this is indeed the brown garden snail, and 80% of you identified it correctly. That is hands down the best result I've seen in any survey that I've done with this species. So that's very, very impressive. Well done. So as I said, this species is called the brown garden snail, sometimes called the European brown garden snail as well. And it is one of the most damaging species we've got on the West Coast. It's a major pest of a huge range of crops, citrus, wine grapes, ornamentals, but it also thrives in most urban and suburban situations. So if you have a snail in your garden back home, if I was a betting man, I would bet that it is almost certainly the species you're dealing with. This snail was first introduced to the United States on the West Coast in the 1800s as a potential source of food. But people never really developed a taste for it, and the snails eventually escaped into the wild. It's also unusual in that this species lays its eggs into nests that it creates in the soil about 2 inches from the soil surface. And when the eggs hatch, the baby snails will remain inside that nest for up to 2 weeks, and that can make it very, very difficult to detect them. Some other damaging pest gastropods here in the Pacific Northwest include the garden slug, which is a small species that tends to be black or dark gray, but it's got a very nice contrasting yellow or orange colored foot. This slug usually lives underground, and it's a very serious pest of vegetables and pasture and even sunflowers as well. I also find it quite frequently at roadside edges, in gardens, in home gardens, and garden centers, I should say, and forest edges and open areas in forests as well. In terms of control, the garden slug appears to be more tolerant of slug baits than other slug species, so it can become a problem quite quickly if it does find its way into a crop. European red slug, and this is one of the largest invasive slugs we've got in the United States. Relatively easy to identify this species because you can see the head and tentacles are a contrasting darker color to the rest of the body. And then the orange foot fringe, it's got these distinct thin vertical black lines. And this slug is found in a whole manner of disturbed areas, and it's got a really voracious appetite for living plants. Surprisingly, it's also unusually common in campgrounds here in the Pacific Northwest. And we think the reason for that is that it's been inadvertently transported on the underside of tents by campers. Also, when it's disturbed or when it's distressed, it often rocks its body from side to side. It almost looks like the slug is dancing. The marsh slug, and this is another common species in the Pacific Northwest. It's a small brown slug with very watery mucus. It's also amphibious, and so this slug does particularly well in the wetter parts of crop fields, and it will happily live underwater for days on end. This slug is also very, very aggressive, and it'll readily lash other slugs with its tail, and it'll happily take bites out of other invertebrates that come too close to it. This is a slug you may see around your home. It's one of the most common species that we find around homes and urban areas. It's called a cellar slug, and it tends to hang out in dark, damp places like sprinkler valve boxes and drainage shores. It's a large yellowish brown, sometimes yellowish green slug. It's got very sticky yellow mucus, and the tentacles are a nice petrol blue color. In terms of food, this slug really likes to eat pet food, would you believe? So you often see it feeding in pets' food bowls after it gets dark. It'll also make its way into people's houses, into trash cans, and feed on trash in people's trash cans as well. An oddity in the Pacific Northwest slug world is this species here. It's called Testacella haliotidia, or the shell slug. And as its name suggests, it's one of the very few species that actually has an external shell. It spends most of its time underground, and it very rarely comes to the surface. And for that reason, we don't encounter it very often. It's also a carnivore, and it feeds almost exclusively on earthworms. And because it doesn't feed on plant material, usually not regarded as an agricultural or horticultural pest. But in saying that, we do not know the impact that this invasive slug is having on potentially beneficial earthworm populations. I've purposely kept the worst to last. The grey field slug is without question the most damaging slug species here in the Pacific Northwest. But not just here in the Pacific Northwest, it's one of the most damaging slugs found throughout the world. It lives in a huge range of environments, and it's a very serious pest of ornamentals, fruits, vegetables, and grain crops, and even specialty crops as well. If you grow or manage plants of any kind, I guarantee you've had this slug at some stage. An easy species to identify, because when it's distressed or if it's disturbed, it secretes a milky colored mucus. And you can see that quite clearly on this darker colored specimen. You can see the milky mucus between the body tubercles. So because the grey field slug is the most damaging species we've got here, one of the things that my team and I wanted to do was to try and understand the life cycle of this species. Because in order for us to effectively control a pest, we've got to know as much as we can about its biology. That's absolutely crucial. So we set up 16 slug traps in a 4x4 grid in 6 annual ryegrass fields in the Willamette Valley. We chose annual ryegrass because it's a very valuable crop here in Oregon, and also it's a crop that the grey field slug does a lot of damage to. This is one of our field sites, and the pink flags here, here, and here, they mark the positions of our slug traps. And these are called slug blanket traps or slug refuge traps. They're essentially a 1/2 meter by 1/2 meter square in what looks like industrial strength aluminium foil on the upper surface. If you flip it over, there's black perforated plastic, and inside there's a highly absorbent material. And slugs really like hanging out underneath these traps because they essentially create a dark, damp environment, and that's the type of conditions that slugs really like. Okay, so what did we find? Well, we brought all of the slugs that we found under our traps back to the lab where we weighed each and every single one of them, because we wanted to determine the percentage of newly hatched slugs, juveniles, adults, and mature adults. In total, we weighed close to 10,000 slugs, and we did find some very nice patterns. So, for example, if you look at the left-hand side of the graph, juvenile slugs, which are in orange, and were by far and away the most common life history stage in October. But as the fall, the winter, and the spring progressed, and mature adult slugs, which are in yellow, became more and more common. Now, there's these slugs, the mature adults, that lay huge numbers of eggs. So it's really important that we control these slugs before they release their eggs into the environment. In terms of slug eggs, most were laid from February through April, and with peak egg laying occurring in early March. So for us, having this sort of detailed biological information is really, really important, because different control strategies have different efficacies against different slug life history stages. So for example, we know that insect natural enemies, so insects like ground beetles, prefer to feed on smaller slugs. So taking advantage of these natural predators during September and October, when juvenile slugs are most common, will likely give us the best results. Also, because the grey field slug lays the bulk of its eggs during the spring in Western Oregon, if we can kill the slugs before they release their eggs into the environment, or if we can control the baby slugs after they hatch from their eggs, then we will have a smaller fall slug population to deal with. Why do I say that? Well, because it's the slugs that hatch from the spring-laid eggs that make up the bulk of the slugs that we see in the fall. Right, so now that we know a little bit about slugs and their biology, I want to talk to you briefly about current control options. As most of you probably know, when it comes to managing slugs and snails, the mainstay is really the use of chemical molluscicides or chemical pesticides. A molluscicide is a chemical that's specific for slugs and snails. And there's a number of active ingredients. Metaldehyde appears to be the most widely used, but it can be very, very toxic to cats and dogs. Iron phosphate, sodium ferric EDTA, these are stomach poisons, but it can take several days for slugs and snails to die. And then we've got metiocarb, but metiocarb is a restricted use material and it's labeled danger, generally only used as a rescue treatment in plant nurseries where infestations have gotten completely out of control. Tillage, and this can also be effective at reducing slug numbers, but with the current drive towards no-till and conservation tillage for soil management purposes, many growers are becoming much more reluctant to use it. Other options include hand picking, the use of traps, and the use of barriers. And while these can be very effective in a small-scale situation, so something like a home garden, for example, and when it comes to larger commercial-scale production, they're just not practical. So I'm going to give you some tips now on best practice methods for baiting. Firstly, the first thing I really want to say is that it's really important to have a uniform distribution of bait on the ground, because this will increase the likelihood that a slug or a snail will encounter a bait and consume it. If a slug doesn't encounter or a snail doesn't encounter a bait, if it doesn't consume it, the bait is just not going to work. Ideal conditions for baiting, temperature range somewhere between 50 and 65 degrees Fahrenheit, high humidity but not raining, overcast, it should be very calm if possible, less than 5 miles per hour wind speed, and moist soil. In addition to using pellets, I would also encourage you to consider using liquid metaldehyde if it is registered in your state, and the product name is Slugfest. It's a contact poison, and you can get very, very good kill rates with Slugfest if you apply it at times when slugs and snails are active. So for example, in the evening or in the early morning. But you should not apply it if there's heavy rain, because the rain dilutes the product and it decreases the efficacy. In terms of monitoring, make sure you pay particular attention to fields which have a history of slug pressure and snail pressure, but do monitor all fields, because gastropods can suddenly become problematic, even in fields which historically have had low numbers. I would also consider rotating baits and active ingredients to mitigate potential effects of bait shyness. And it's important to remember that broad spectrum insecticides can kill natural predators of slugs and snails, so insects like ground beetles, for example. And this decrease in natural enemy pressure can cause a really significant and sudden increase in slug and snail numbers, even in fields which in the past have had low numbers. And minimizing weeds, this is always good advice, and good advice that I like to give, because reducing the number of weeds reduces alternative food sources for slugs, and it reduces reduces shelters. And so essentially by removing weeds, you're making fields less hospitable for these pests. Any form of soil disturbance, be it ploughing or minimal till, also decreases slug shelters, and it brings slug and snail eggs to the surface, where they dry out and die, or they're fed on by predatory insects. And lastly, important to remember that open, damp, and cloddy seed beds provide ample shelter for slugs and snails, and that generally leads to very high populations, and in general, then that leads to significant crop damage. So I've got some more questions for you folks. I want to learn a little bit about your molluscicide use, your slug and snail control methods, and so on.
So before we do the poll really quick, there is a question, and I think it may have gotten answered, but I wanted to pose it to you just in case you had something else to say about it. The question is, which of the chemical options are OMRI listed for organic?
So the only chemical control option that's OMRI listed is iron phosphate. Iron phosphate is approved for use in all organic systems, and the other ones are not. Talking to growers throughout the US, the perception among growers, I would say, is that iron phosphate does not perform as well as metaldehyde, for example. And the work that we've done in my lab suggests that both can be very, very effective, but the mode of action is very different. And metaldehyde paralyzes slugs and snails. And so if a grower returns to a field that they've baited, if the bait has worked, they see lots and lots of dead and paralyzed slugs and snails in the field. Iron phosphate has a different mode of action, as I said. It makes the slugs very, very sick and the snails very, very sick. So they tend to crawl away and hide. So you don't see that same level of mortality in the field if you use an iron-based bait. But that does not mean that the bait is not working. If you're using an iron-based bait, it's better to look at crop protection as opposed to looking for dead slugs and snails.
Okay, perfect. So I'll go ahead and launch question 6, and there might be some more questions that come up after that. Can you all see that poll question? Which of these active ingredients do you use most often? Leave it open for a few seconds here so everybody has a chance to answer. Okay, we'll go ahead and end the poll. And you should be able to see the results.
That's really, really interesting. Usually when I run this poll, the vast majority of people that take the poll say that they use metaldehyde. This is the first time that iron phosphate and Sluggo has topped the poll significantly. So perhaps many of you are organic farmers or farming in urban areas where metaldehyde poses a risk to dogs and cats. That's very interesting. Maybe something we can discuss after my presentation.
Yeah, for sure. Okay, so we're going to go to question 7. 7. Should be able to see now. How satisfied are you with performance of slug and snail baits? Okay, looks like majority have answered this one. Go ahead and share those results.
Interesting. So again, most people, when I ask this question, they're on the neutral side, so neither satisfied or unsatisfied. That appears to be the general pattern here. 32% of you are satisfied with performance, about a third, and about a quarter are unsatisfied, with 8% very unsatisfied. Okay, that's very useful information for me. Thank you.
Okay, and number 9. How much approximately do you spend on slug and snail control per acre each year? Did I skip number 8? I'm sorry, Rory.
I think you may have, but don't worry, Jess, it's okay. I mean, this is a kind of a similar question. So for this one, if you consider the use of molluscicides and time spent monitoring, time applying baits, crop destruction, reseeding, and so on.
Looks like most folks have taken this, so we'll go ahead and end this one and share those results.
Okay, great, thank you. So most of you less than $25 per acre, some about a third as much as $50 per acre, and 4% spend $100 to $500 per acre. Wow, in slug control, that's a lot of money. Okay, great. I think the work that we're doing in my lab on developing novel approaches is going to be particularly relevant to lots of folks attending this evening. So thank you for all that information. That's very useful information for me to have. Is there any more questions, Jess? Do you want to ask some more? If there's any more questions, I'm happy to take them now, or we can wait until the end.
Looks like we did have one that came in. Well, two actually. So in comments to Sluggo, stopped using it because the birds are eating it. They don't feel that it can be a good combination of chemicals for birds. So that's, I don't know.
Yeah, I only would have to say I don't know what the impact of eating Sluggo will be on birds. It's the active ingredient is iron phosphate, and then the inside the pellet, as well as the iron phosphate, there's an attractant. It's generally a flour base. It's generally flour and inert ingredients. But I don't know. I do not know what the impact would be on birds. I don't know if there's any scientific literature on that. But that is certainly concerning that birds are picking up the pellets and eating them. And if it's most slug bait and snail baits now come in different colors, there's a blue option and there's also a brown option. And most companies recommend that if non-targets are eating your bait, you go with the brown option because it's more difficult for animals to see it. It's much easier to see a blue pellet in a field than it would be for a brown pellet, for example.
Mm-hmm. There's a couple other questions about Sluggo that you might want to answer now since we're chatting about it. So one person is asking, should they put out Sluggo in mid-September? They read that they should put it out in mid-September to kill slug eggs. Does this work? And the second kind of, someone else asked a similar question, is Sluggo effective against baby slugs?
That's a very, very good question. And I would say that the jury is possibly out on that. Work we've done in my lab shows that baby slugs are reluctant to feed on any pellet that we provide to them. And we don't know the reason for that just yet. When you look at the literature, some folks say it's because their jaws aren't developed well enough to be able to. So slugs, essentially, if you look inside their mouth, they have a very sharp beak, and they use that beak to scrape food into their digestive system. Some folks say that their beak is not developed enough to be able to scrape the pellet into their mouth. But in terms of controlling baby slugs, we find, at least here in Oregon, using the liquid metaldehyde product is very, very effective. It doesn't look like metaldehyde is an active ingredient that you as growers like to use, so it may not be an option. So yeah, the jury is out on whether Sluggo would work for baby slugs or not. In our lab, it suggests that it doesn't work very well, unfortunately.
Would you have any suggestions on timing to use Sluggo effectively?
Good. So generally, it's recommended to use Sluggo in the early fall. So I think, as the person asked the question said, we recommend its use in September before the heavy rains arrive. And when the heavy rains arrive, we recommend the use of more rain-fast molluscicides, something like Metorex, which is again a metaldehyde-based bait. And then Forox AQ is another one. And I don't recall whether the active in that is iron phosphate or sodium ferric EDTA. If it's sodium ferric EDTA, it's not approved for organic production, but sodium ferric EDTA has a lot less non-target effects than metaldehyde, for example. So using iron phosphate, better to use it in September before the rains arrive.
Yeah, okay. And then one last thing on Sluggo. Seems to work on slugs, but the brown snails seem to ignore it. Why is that?
Good question. We've actually found about 2 months ago, we got a small amount of money from a company to test all of the different molluscicides, metaldehyde, iron phosphate, and sodium ferric EDTA against European brown garden snail. We just did it in the lab in a simple experiment in big plastic containers with soil. We found that baits that had sodium ferric EDTA as the active ingredient were most effective against the European brown garden snail. We don't know why, and it's probably something to do with the attractiveness or the palatability of the bait. There's some evidence to suggest that slugs and snails develop what's called bait shyness. So if you use the same bait, the same process, for example, if you were to use Metorex and only Metorex, there's some evidence to suggest that if a slug or a snail consumes a pellet but doesn't die, if it takes a sublethal dose, it will avoid pellets of Metorex in the future. So that's why I said in one of my recommendations, it's good. It's good advice to mix up the active ingredients you use and the different products that you use to avoid against potential impacts of bait shyness. So I don't know why certain slugs and certain snails prefer one active ingredient over another. It might be to do with the actual palatability of the bait.
Okay. And there's another one in chat, but I think I'm going to hold that question over until your next part because it's about using wheat flour, water, and yeast or beer traps. I think this is a good segue into that.
Okay, excellent. Okay, thanks, Jess. Okay, so I was hired by Oregon State University in 2016. And when I was hired, I was charged with developing new tools for managing slugs and snails. And that's really been the focus of the research in my lab over the past 6 years. And we've been investigating a wide range of new approaches for managing these pests in the Pacific Northwest. And today I want to share with you what we've discovered and where we are at certain stages of this novel control research. So I'm going to talk to you about novel attractants and also biological control using natural enemies. For a long time, I've been very interested in developing novel attractants for pest gastropods because there's really lots and lots of information when you look through the scientific literature that suggests that chemicals play a very important role in a whole host of slug and snail behaviors, so everything from feeding to predator avoidance to reproduction. One area where I felt we could get some solid results quite quickly was in the identification of a highly attractive food source. And I say that because we know that certain gastropod species are attracted to the odors or extracts of certain plant species. So for example, the European brown garden snail is known to be strongly attracted to the odor of carrots. So we were curious to learn if we could exploit this behavior to help us detect and potentially manage slugs and snails. When most people think about gastropod attractants, the very first thing that comes to mind is beer. And beer is a pretty effective way of attracting slugs and snails. But surprisingly, not much work has been done on investigating other fermenting products as potential novel attractants. And despite other fermenting foods being used to help control other pests, things like cockroaches, for example. So my team and I, we decided to try and fill this gap, this knowledge gap, by comparing the attractiveness of a whole bunch of different fermenting foods in the lab. We conducted several hundred tests and we identified one food as being consistently the most attractive. And that food was this. The bakers among you will recognize it immediately. It's a basic Bredo mix of flour, yeast, and water. Now, this work has gotten lots and lots of press attention. And we've had articles in the Corvallis Advocate, in the Columbian, the Register Guard, and the Oregonian. And even in USA Today, our Slug Traps were in a headline in USA Today in September of last year. Recently, there was an article on Bredo as an attractant for slugs in the Canberra Times, which is a newspaper in Australia. And the Australian Department of Agriculture are now using Bredo as a novel detection tool for new invasive species coming into Australia. So I think this sort of press coverage, it really speaks to the need for the development of novel slug control tools for growers like yourselves in the Pacific Northwest, but also growers throughout the United States and growers throughout the world for that matter. After making the discovery, one of the things that we needed to do, and it was something that was very important, was to test the Bredo in the field. It's all very well that Bredo performed well in the lab, but the true test comes when we trial something like this outside. So we set up 20 traps, and you can see one of the traps here. And we had 20 traps with Bredo and an additional 20 traps with just water. And we placed them in a perennial ryegrass field here in the Willamette Valley. And we knew the field was infested with great field slugs. We did pour some liquid metaldehyde into the traps because we wanted to be sure any slugs attracted into the traps during the course of the night would be killed. And as you can see in the graph, after just 24 hours, we collected significantly more slugs with the Bredo compared to the water control. In fact, we collected an average of 90 slugs with the dough per trap compared to just 20 with the control. And that was just over a single night. And we did also test how long the dough remained attractive in the field. And our data clearly shows that it's attractive for at least 8 days. So it does have a very good field life. At the minute, we are working with a chemist in Canada, and we're trying to recreate the odor of Bredo using chemicals in his lab. Now, you may be wondering, why are we trying to do something like that? Well, the reason is that if we can successfully recreate the Bredo odor, then we will have much more control over our attractant. So we'll be able to manipulate things like concentration and release rates. So essentially, we'll be able to tweak our attractant to make sure that it is as attractive as possible to different pest gastropod species. And also importantly, having a synthetic odor blend would make it much, much easier to incorporate our attractant into existing baits. And that would make them more attractive and ultimately more effective. So Jess, I don't know if you want to ask that question now that was in the chat about kind of Bredo mix.
Yeah, so one of the participants asked, what about wheat flour, water, and yeast or beer traps?
Sure. So we compared lots of different beers in the lab when we were doing fermenting, comparing the different fermented products, and different beers had different levels of attraction, but Bredo was by far and away the best attractant that we've ever tested. You know, as it stands, it is definitely a useful tool for trapping slugs and snails in a garden scenario. If you've got a large crop field, it's not, in my opinion, it's not very practical to go out there with Bredo and try and trap or collect all of the slugs. And maybe there are some folks who will be interested in doing that. That would be great. But it was one of the reasons why we want to develop this synthetic Bredo blend, so that we can have more effective baits. But in a home garden scenario, it works extremely well. I use it in my garden to trap slugs and snails. In my garden, I make the Bredo, I roll it into small balls, and I leave the balls out in the evening time. I come back after about an hour, a couple of hours, and then I just collect the slugs and snails that I've gathered around the dough balls, and that works very well. And for those who are interested in using it in a trap and don't want to use a toxicant with it, you can make a more watery Bredo mix. At the minute, we use 500 grams of flour, 500 milliliters of water, and I believe it's a quarter of an ounce of activated dry yeast. And you just mix those together and it works really, really well. But if you make a more liquidy dough, for example, you could use more water, and you could put that into a small container and put it into the ground like a bear trap. So it would work similar to a bear trap, but it would be much more effective at trapping slugs and snails. Okay, if there's no additional questions, I'll move on. Another tool that I'm really interested in developing here in the Pacific Northwest is biological control. And for those of you who don't know biological control, it's the use of a natural enemy to suppress a pest population. When it comes to slugs and snails, this is a very, very under-researched area in the United States, despite the fact that slugs and snails have a diverse range of natural enemies, and a commercially available biological control agent is currently being used throughout Europe to help growers manage slug and snail pests in a wide range of different crops. That product is called Nemaslug, and the active agent is a nematode called Phasmarabdites hermaphrodita. You don't really need to remember the name, but it's lethal to a wide range of pest slugs and also some snails. The product itself consists of about 12 million infected nematode juveniles mixed with clay. Very simple to use it. You essentially mix the clay with water and then you apply it to the soil in your target area. Nice thing about Nemaslug is that it is only lethal to slugs and snails, and it can be very, very effective. So for example, in the United Kingdom, slug damage to winter wheat has been reduced by up to 89% when Nemaslug has been used. And in the Netherlands, gastropod damage to orchids is consistently reduced by over 95% when this product is used by growers. When I talk about this nematode at outreach and extension events, the first question I'm always asked by growers is, where can I get my hands on some of this product? And unfortunately, I've got to tell them that they can't. Nemaslug is not available in the United States because up until recently, the nematode had never been found there. But 8 years ago, when I was working in California, I traveled all around the state collecting slugs and snails to screen for these nematodes. And we ended up finding the European species Phasmarabdites hermaphrodita in multiple locations. And this was the first time that the nematode had been found in North America. So it was a significant discovery. Then I moved to Oregon State University, and finding this nematode became one of my top priorities. Over the past 5 years, and my collaborator, his name is Dee Denver, he's a professor here as well. We've been conducting surveys throughout the state in a wide range of different crops. And to date, we've collected about 5,000 slugs and snails from 50 different sites. And we bring them back to our lab, and then we keep them in plastic containers, like you can see in the slide. And these containers have damp paper towel and just some organic carrot. And we check the containers every other day, and then if we find slug cadavers, we place them under a microscope and we look for nematodes. So this is what a nematode infested slug looks like. You can see it's a pretty gruesome sight, and there are hundreds and sometimes thousands of nematodes crawling all over the slug's body. When a nematode finds a living slug, it crawls up onto the back of the slug and it penetrates the body through a small pore at the back of the mantle. And once inside the slug, it vomits up a bacterial soup, and that bacteria release a toxin that kills the slug. And then the bacteria reproduce on the decomposing slug flesh, and the nematode feeds on the reproducing bacteria. At this stage, we typically take individual nematodes from the slug carcass. We extract DNA, and then we use that DNA to identify the nematodes. What have we found? Well, here in Oregon, according to our genetic analysis, we found a total of 4 species, and all 4 seem to be restricted to the western part of the state. The first, Prehermaphrodita, remember that's the species that's been commercialized in Europe and that growers are currently using throughout Europe to help manage slugs in their crops. We found that in Corvallis on the main OSU campus, and also just outside Salem in a field of brassica. Phasmarabdites californica, and this is another species. It appears to be the most widespread Phasmarabdites we've got in the state. We found it in Brookings in the southwest corner of Oregon, also in Eugene, in Corvallis, and in Portland as well. P. papillosa, this is another European nematode species that is known to be lethal to slugs and snails, and we have found that in Portland. The last species is a species that's new to science, so very, very few people have seen it, and it does not have a complete name just yet. And we found that in one location outside a small town called Malala. So although we have found 4 different nematode species, and we did decide to focus our efforts on just one, Prehermaphrodita, which is a species that's commercialized in Europe, so it was an obvious choice. And also we also had limited funding, so we didn't have resources to look at all 4. The next logical step then in our research was obviously to try and figure out if our US strain of Prehermaphrodita was lethal to key gastropod pests. And thankfully, we were able to grow it in sufficiently large numbers to test its lethality in infection trials. To run the trials, it's very, very simple. We set up these containers which are damp sterilized soil. Some of the containers had no nematodes, these were our controls, and some were inoculated with 20,000 nematodes. We then added our target slug or our target snail. We replicated the containers 5 times, and then we came back every day and we recorded slug mortality. So our first target was obviously going to be the gray field slug, which is that slug that's causing the most damage to crops in the Pacific Northwest. And as you can see from the graph, the US strain of our nematode was highly lethal to this species. So on our y-axis, we've got the mean percentage slug mortality. X-axis, we've got the number of days after the slugs were exposed to the nematodes. As you can see, after just 4 days, slugs had started to die. By day 6, mortality was up to 40%. And by day 8, it had increased to 80%. And after just 12 days, all of the slugs in all of the nematode containers had died. Next up, we tested it against the white-soled slug, which is a very damaging pest of grass seed production in the Willamette Valley. Now, in general, I would say roundback slugs like this species are more hardy and much more tolerant of control strategies than the gray field slug. But again, as you can see from the graph, the white-soled slug is also clearly susceptible to our nematode. Again, after 4 days, specimens had started to die, and there was continuous daily mortality up until day 19, when all of the slugs in the nematode containers had died. Next slug is the marsh slug. Again, remember, you may remember that this was the species that I said is a very serious problem in wetter crop fields. Species or specimens started to die after just 2 days, so very, very quickly. And after just 9 days, all of the slugs were dead in the nematode containers. So this slug succumbed very, very quickly to our nematode. After running these experiments, then we were able to conclude that our US strain of P. hermaphrodita is highly lethal to 3 of the most damaging slug species of agricultural production in Oregon. Over the past months, we have found that the nematode also kills other invasive slugs and snails, so the European red slug, the shell slug, juvenile European brown garden snails, and even juvenile giant African land snails, which is one of the largest and most damaging snail species in the world. The next step then was to test the lethality of the nematode in a microcosm study. So a microcosm is essentially, like a small field scale experiment. And for our study, we used these plastic containers that we planted with annual ryegrass. And we had 2 rows that were separated by about 5 inches, and we had 100 seeds in each of the rows. We then added 10 grey field slugs to each container when the bulk of the plants were in the 2-leaf stage. And then we treated the containers with either our US strain of the nematode at the recommended application rate for Nemaslug, the European product, or just water. And water was our control, obviously. We had 5 replicates, and then we recorded slug mortality every single day. So let me show you now what we found. So we've got mean percentage slug mortality on the y-axis, number of days after exposure to the nematode on the x-axis. Nematode data is in green, and our control data is in blue. So as you can see, the microcosm data confirmed the results of the laboratory infection trials that we ran. The figure clearly shows that the US strain of our nematode is highly lethal to the grey field slug in a microcosm setting. All slugs were dead after just 9 days. So far so good then with this research, but the next step is going to be really, really important because we need to test the lethality of the nematode against native slugs such as the banana slug and native snails, so Pacific sideband snails, for example. And if we can demonstrate that these and other native slugs and snails are not harmed by our nematode, the pathway to commercialization should be easier. If they are susceptible, then that's obviously not good news. But we do have 3 other species that we can fall back on if necessary. So all our eggs are not in the one basket. Okay, some final questions for you guys before I wrap it up.
Okay, so we're looking at question 10. And the question is, if novel tools such as biocontrol were available for slug snails, how likely are you to use them if they were the same price as current methods? Looks like we're still getting a few people answering. Okay. Go ahead and end the poll.
Yeah, I'm not surprised most of you said you would try. It's a bit of a no-brainer, I think, that you would try this tool if it is proves to be safe. Okay, great, thank you.
Question 11 is, if these tools were more expensive than current options but gave more reliable control, how likely are you to use them? Okay, looks like majority have answered.
Okay, so again, more or less the same reply. Most people are willing to try, and some would wait for others to try first. Some, when 4% said they would not try, then maybe that's something we could discuss at the end. I'm curious to know why some growers would not be willing to try it. Okay, great.
Yeah. Question 12. Let's see, if these tools consistently controlled slugs, how much extra would you be willing to pay? So there's a couple options there. You might have to scroll on your screen to see some of them on the bottom.
And the reason I asked this question, in Europe, the Nemaslug product is more expensive than current control strategies. So if this does become a viable tool in the Pacific Northwest, it would be more expensive than just using baits, for example.
Yeah. Looks like we've got a few people still answering. Right at that mark. So we'll go ahead and end that one.
Great. So yeah, when I typically ask this question, most of the folks who reply say they'd be willing to pay either 1.5 times as much or twice as much. Very rarely does someone say 5 times as much, but it does happen typically in high value crops like ornamentals and so on.
Yeah.
So thank you. That's good to know.
Okay. Question 13. Back to the what is this slug?
So I'm curious to know if there would be a change in the amount of folks who, Jess, I can't remember what the first, when we asked this question first, what there were, how many, was it about 25%?
I want to say, yeah, we can look back after.
Okay, great, we can check later. Great, thanks.
Let's see, I think most folks have chimed in on this one, so go ahead and end that poll.
Oh,
You did your job.
I did my job. Good stuff. Actually, that's amazing that 100% of people got it right. Typically, it gets up to about 70 or 80%. So this is obviously a very attentive audience. So that's awesome. Well done. So the next one, I'm going to ask you what this snail is again, but I was super impressed with how many of you got it correct first time around. And so even there's just no improvement, it's I think it was almost 80% the last time. I think it was 88% in fact. So let's see if there's any improvement.
Let's see if anybody else is going to answer here. I think it's pretty
100%. Well done. This is hands down the most attentive audience I've ever presented to. Very well done. And the reason why I bring these two species again photographs is I'm trying to drive home the point that it is really critically important that we know what pest we're dealing with in order for us to develop the best management plan for that particular pest. So for example, gray field slugs are a lot more susceptible to the use of baits than slugs in the genus Arian, so the white salt slug, for example. So it's really important that you know as a grower what pest you're dealing with in order to be able to make the best informed decision about how to control it. And so I'm glad to hear that your identification skills have improved.
Yeah, so to answer your question on the first one, the gray field slug was answered at 28%. So you're
wow. Okay, excellent. So there may be some more additional questions, but I just have 2 or 3 more slides. I think we'll just go through those and then we can maybe ask the rest of the questions and have some discussion if that's okay, Jess.
Yeah, we're good.
Great. Okay, so for those of you interested in learning more about slugs and snails, I did put 2 resources on this slide. The first one link will take you to a free guidebook to the slugs and snails of Oregon. You may wonder why it's Oregon and not Washington. I don't know if there are any slugs or any guides to slugs and snail of Washington State, possibly not. But you up in Washington have the same slugs and snails as we do in Oregon. So this guide is directly applicable to your state as well. The Slug Portal and this second link, if you type Slug Portal OSU into Google, it will be the first site that comes up. This is a website that I helped to build. It's got tons and tons of information on slugs, how to identify them, how to monitor for them, and information on their biology and ways to manage them as well. So these are very good places to start. I do want to thank US Department of Ag, Oregon Department of Ag, California Department of Agriculture, European Union, Oregon Sea Council, and Oregon Association of Nurseries for grants which fund my program. Andy Colton, Chrissy Dodge, and Casey Richard are scientists who work in my lab. I'm very, very lucky to have them working for me. They do exceptional work. I'm not going to go through each of these folks here. These are key collaborators for my program, and without their involvement, we would not be able to get all the work we do done. So I'm very grateful for their collaboration. Also, big thanks to all of you for joining us this evening. I know this is a very busy time of the year for you, so I'm grateful that you took the time to listen to what I have to say. And do know that if you have any slug and snail questions, you can contact me. That's my email address, rory.mcdonnell@oregonstate.edu. I always enjoy hearing from growers and trying to assist whenever I can. If you're into social media, I'm on Twitter. I'm not very good at social media. I'll hold my hands up, trying to get better. But if you're into social media and you want to connect, you can connect with me at Rory J. McDonald. Thank you very much.
Thank you so much for that awesome presentation. I think this has been, I shared with you before, this has been a topic that's been on our list for regional small farms for a while now. So I'm so happy that we were able to engage you in giving this talk. But I do want to open it up because I know we do have some questions that are in the chat, and I'm sure there's going to be some more coming through as we're answering some of these. So I'm going to just start from where we left off so that we're not missing anybody. So there's a question about how do you dispose of the snails and slugs once you trap them? And I think this was back when you were talking about when you're trapping with your bread
Sure.
dough. So
the number of ways to do it. Probably the most humane way. Well, it's difficult to know if there's a humane way, to be honest. What a lot of home gardeners do is they have a bucket of water that they put some dish soap into, and that breaks the surface tension. So if you drop the slugs and snails into that container, they will drown. Others put them into a Ziploc bag and freeze them, and then dispose of them in the regular trash. So something like that tends to work best. That's what home gardeners use. For our work, we tend to bring the specimens back to the lab if we're trapping them alive, because we're always in need of slugs and snails for experiments in the lab.
Yeah. The next question is, does sourdough also attract slugs?
Yes, it does. Any fermenting bread dough mix will attract slugs, and that raises an interesting question. And one of the things we have done relatively recently is we've tried different flours, different yeast to see if we can improve the attraction of our dough mix. We do get some small differences in attraction, but different flours and different yeasts tend not to have a significant effect on the overall attraction of our mix. So we just use a very simple enriched white flour. In fact, I should really have put the recipe in the presentation, so my apologies. What I will do is I will send the recipe to Jess tomorrow, and she can send it out to folks who are interested. But we buy Kroger brand enriched white flour, and we mix it with water and active dry yeast. So very, very simple mix. We've also tried adding sugar to the mix to see if it would make it more attractive, and it didn't. One of the things we were curious about was, obviously there's limited food for the yeast to feed on. And we wondered that when the yeast break down all of the flour, could we recharge the mix by adding a cup of flour, for example? And it looks like you can. So just by adding flour to the mix appears to continuously make it attractive. If you are going to use bread dough, when it dries out, it starts to lose its efficacy. That's why we use the bread dough in the traps, in the snailer traps that I showed you, those green capsule-shaped traps, because it protects the dough somewhat and prevents it from drying out. And so it's good to protect the dough somewhat if you're going to use it.
That's great. So I think this question, going back to your research with the nematodes, your most current stuff, what's the life expectancy of the nematodes? Do they need to be replenished each year?
That's a very good question. So once the nematodes are applied, if there are sufficient slugs in the field, they will continue to maintain very, very large numbers. And once the slug population decreases, the nematode population will decrease in tandem as well. So yes, the nematodes would need to be applied on a yearly basis. They do not disappear from the field completely once you apply them. And we have found in some persistent study work that we've done, when we apply them in these microcosm settings, after all the slugs have died, we're still detecting the nematodes in the containers up to 99 days after the last slug has died. So they are persisting in the soil, and they're probably, these nematodes, although their preferred host are slugs and snails, they can also survive quite well on just decomposing organic material in soil. And we suspect that that's what they use to remain in ecosystems once they invade them. But if there are slugs and snails, that's definitely their preferred host. But they would need to be reapplied on an annual basis. We're pretty sure of that.
So kind of a follow-up question to that, along those same lines, are you looking at the effects of the biological controlling nematodes on the crops as well as killing the slug?
Yes, we are. Great question. So initially we started out by just, the first question we wanted to ask was, well, are the nematodes here first of all? And we knew that. Now we can say that they are. Second question we wanted to ask, well, do they kill the key slug and snail pests? We know yes for that. And then recently we did some crop protection studies as well, not in the field. We're not going to do any field trials with these nematodes until we're sure that they're not going to have any impact on non-target species. So all our work is done in secured greenhouse facilities. And we have found that these nematodes do protect the crop, but it takes longer to see crop protection with a nematode than it does with a pesticide. So our data very clearly shows if we use liquid metaldehyde as a treatment in our microcosm studies, we see immediate crop protection with the metaldehyde. With the nematodes, it takes 3 or 4 days for us to see the crop protection element coming in, because the infection process takes a while with the nematodes. They enter the slug and then the slug gets sick. So it takes 3 or 4 days for the slugs really to get so sick that they're not going to start feeding. So one of the experiments we're doing at the minute is we're looking to see if we can use molluscicides or pesticides with the nematodes as an IPM approach. So for example, can we use a molluscicide to get that crop protection early and then use the nematodes to get sustained crop protection after the effects of the molluscicides wear off? Because we very clearly see when the effects of the molluscicides wear off, if the slugs aren't killed, they will damage the crop. But the nematode is so effective at killing the slugs that you get long-term crop protection using the nematode. So if we can use both the pesticides and the nematode, then we've got best case scenario. And I can share with you today that metaldehyde, iron phosphate, and sodium ferric EDTA appear to be completely harmless to these nematodes. So it looks like they can be used together as a crop control tool.
That's great. One other question about brown snails. What controls can be used and still leave the brown snails edible?
That's a very good question, actually. I don't know if there's any, to be honest. Typically when we use controls for brown garden snail, we're trying to kill them. So I guess the only thing you could do would be to try and attract them to the Bredo and then pick them off from the Bredo. We do know that the brown garden snail is very attracted to the dough. So you could put dough balls out into your yard, and as the brown garden snails come, then you could pick them off. And just as a side topic, if you find that Bredo is not working very well for you, I would be very surprised if it doesn't. We found that it works against multiple species from temperate regions of the world, but also from tropical regions of the world as well. But a good backup to Bredo is chopped cucumber. There appears to be something inherently attractive about chopped fresh cucumber for brown garden snails and grey field slugs. So about 3 years ago, I was touting cucumber as the best novel attractant you can find, but I've had to eat my words since because Bredo far outperforms it. But if you're found for some reason the Bredo is not really working for you, you could try chopped cucumber. There's just something about it that attracts slugs and snails as well.
That's really good information. I had not heard that before, actually. So another question, why add flour and not just the yeast?
Because yeast on its own is not as attractive. We use the yeast that we use is not activated. And so if we just put yeast out, we really don't get any attraction. But you can also turn that question on its head and say, why don't you use just flour and water? Again, because it's not attractive. The magic ingredients are when you put all three together. And we're confident that it's some volatile component that the yeast is giving off while they're basically fermenting the bread dough. So as I said, when we're talking about slugs communicate by chemicals. In our world, we perceive our environment through vision and through sound. Slugs have very, very poor vision. They can detect light and they can detect shade. We've got some data to suggest that they can detect certain shapes, but they don't have any auditory system. So in their world, they perceive the world through chemistry. And we're as certain as we can be that it's some chemical being released by the yeast when they're breaking down the flour that the slugs and snails are homing in on. We don't know what that chemical is yet. And as I said, we're working with a chemist in Canada to try and figure out what that is. And hopefully we'll be able to figure it out.
Yeah. So along the same lines with the yeast and the bread dough, the next question's about when you were talking about using your dough balls, do you put them beneath a cover? And if so, what kind of dimensions and what do you recommend to use for the cover?
So this is what we put them in. So this is called a Snailer trap. And you can see it's a very, very simple trap. It's just a piece of plastic with a capsule shell. And then the way it works is that the snails can go into the trap by putting these prongs, but then they can't go out because the prongs are slightly longer than the trap itself. So you can see these will open in like that, but you can't pull them outwards. And these traps are available from a company called Acre Organics. I can send you the link, Jess, tomorrow, and you can send it to the attendees. And they're about $7 each. If you're not willing to spend that much money, you can essentially make one of these yourself, to be honest. You could just get a large plastic yogurt container and cut a couple of holes in it and then put the dough ball inside. But just know that the slugs will be able to go in and snails will be able to go into the container, feed on the dough, and then escape as well. So yeah, it is important to keep the dough covered in some sense, particularly during the summer. Once the weather cools down and gets wetter, then it's not so important.
Yeah. So going back to the nematodes, what time would you apply them or reapply them, I guess, each year?
So here in Oregon, I would say in grass seed production, which is a crop that I do a lot of work in, growers here, well, let me backtrack a little bit. So in the Willamette Valley, this time of the year, it is extremely difficult to find a slug if you go into a field, unless the field is irrigated. And we enter kind of a drought scenario. I'm not sure if it's the same up in Washington where you folks are based, but it's almost impossible to find a slug. But then once the fall rain comes, typically once we get 2 inches of rain, the slugs start to come up from the soil where they've spent the summer in a kind of a summer hibernation phase. But once they start to feel the water percolating down through the soil, they're going to go up to the surface and start feeding. So once there's about 2 inches of rain, that's when we advise the growers to start putting out their slug control products. They're just baits at this stage. And so it would be the time to apply the nematode. Should also state that this nematode is dependent on a moist soil. If the soil dries out, the nematode will not survive, it will die. Also, it's pretty sensitive to UV light. And so during the summertime, the nematode would disappear down into the soil as well. But that's all good because the conditions that the slug likes is the same conditions that the nematode likes, and that's what you want. The last thing you want is conditions that the nematode likes that are completely different to what the slug likes, and that's not going to work at all. But the two of them require the same sort of environmental conditions to be active. So if this nematode does become available, I would recommend applying it when you anticipate slugs to be a problem.
And then there's one last question right now in the chat, and I just want to let the attendees know we're going to stay on as long as you want, but Rory has generously given so much time of his, and he's got to hop off here in a minute due to some other stuff. So if we do have to have you hop off, Rory, I just want to say thank you so much in advance for being with us. But just know that Laurel is on, and I don't know, Laurel, if you want to give a wave or say hi. So for those of you who are in Clallam, Jefferson, or Kitsap, Laurel is our IPM regional specialist. So she is available to farmers in our 3 counties for technical assistance. She's able to come out if you have a problem that you need identification assistance on, or you want assistance with management techniques, resources. She's the person that you would be going to. So I wanted to make sure that everybody is aware that Laurel's also on the call. And so if there are people that have additional questions after this or want to chat, we'll still be on. But Rory, I don't know if you want to answer this question, or Laurel, maybe this is kind of a general question. I find large brown slugs in the compost bins and their eggs. I feel that I'm spreading them with the compost. Do these types of slugs eat living plants or decaying matter? The brown slug you mentioned are much smaller.
So the interesting thing about slugs and snails, if you look at slugs and snails in general, the vast majority of them have a preference for decomposing plant material. And we know that because when you work on slugs and snails, the color of their poo tells you what they fed on. So for example, if you feed a slug or a snail just carrot, their poo will be orange. If you feed it lettuce, it'll be green. I would say 99% of the time, the slugs and snails that we collect in the wild and we bring them back to our lab, their poo is brown. which indicates that they're feeding on decomposing plant material or organic material in the soil. So without question, slugs and snails in general prefer decomposing plant material. But if you put them in a cropping system and there's not a lot of decomposing plant material, they're going to feed on the crop as well. And don't get me wrong, they like to feed on green plant material as well because it provides additional nutrients to them. So to get to the question, if you're in your compost bin, they're definitely very happy feeding on your decomposing plant material. Possibly if you're spreading your compost, you could be spreading the slug eggs for sure. If you're saying it's a large brown slug, I suspect that's probably the European red slug. Name can be a little bit deceiving. It's not always red. It can be orange, it can be brown, it can be cream, it can be black. But it does sound like it might be the European brown garden snail. So yeah, they definitely feed on decomposing material. One thing that I do want to say before I sign off is I'm surprised nobody has asked me why have we found the nematode that we're researching in Washington. I was certain that would be a question I was going to be asked. I didn't get time to put it into the talk because we've only learned just recently. I believe it was August of last year, we surveyed in Western Washington. We started in Vancouver, we made our way all the way up to Bellingham. And we were confident we were going to find a nematode because we found it in California, we found it in Oregon, and it would make sense that it's in Washington as well. But to our surprise, we did not find it. So, but I didn't give up. In May of this year, I decided to go back to Western Oregon and to Eastern Oregon collecting slugs and snails. And when I brought them back to the lab, we have found the nematode in Washington. We found it in Vancouver and we found it in Spokane. But it's not the European species Hermaphrodita, it's the species that we call Californica. But if Californica is there, Hermaphrodita is almost there as well. And we do have plans to do some more surveying in the state to try and find what other species are there as well. And so I just wanted to let folks know that it is there. It's almost certainly there, and we have found one of the species.
That's great. That's a great update. Thank you. I didn't even think to ask you that, so that's good that you brought that up. I'm sure some other folks might have been having that in the back of their head. Well, I want to say thank you, Rory. I know you have to hop off. Thank you so much. We really appreciate your time and all your knowledge on this matter. And thank you for being available to our farmers who are interested in connecting with you if they have specific questions.
Great. Thank you, Jess, and thank you everybody again. It was great presenting to you. As I said, feel free to reach out if you have any slug and snail queries. I'm very happy to help, and good luck with the rest of the summer. Thanks again.
Bye.
Take care, everyone. Bye-bye.
So for those of you, if there are any other questions, like I said, Laurel's on here, but I do want to point out, we'll be sending out the slide deck for this, as well as a link to our video recording, which will be housed on our online learning page for Regional Small Farms for future reference for anybody who didn't attend or wants to relook at some information, and then a resource sheet with some of those links that Rory had mentioned, as well as his contact information. So I'll make sure, and then the recipe, obviously, for his Bredo and what they've been doing. Laurel, do you have anything that you want to say before?
No, in fact, I think this is one of the most unique and informative presentations I've seen on slug management. And I love that cucumber idea. I mean, everybody has overripe cucumbers at some time of year, so put them to use. Yeah, I enjoyed that probably as much as everybody here. And anyway, yeah.
Awesome. Well, again, thank you guys for joining us. We'll send out the materials tomorrow, hopefully. It'll also include an evaluation. It'll be a mixture of some WSU questions and some OSU questions, since Rory is an OSU Extension specialist. And hopefully you'll be able to take that and give us some good feedback. And join us for our next Dirt Talk. Our next Dirt Talk will be actually in person, or I'm sorry, no, our next Dirt Talk will be online on agritourism. That's been rescheduled for August 23rd. So if you haven't signed up for that and you're interested in that topic, please go online to our website and sign up for that. And then the one after that one is actually going to be a Dirt Talk Farm Walk, which is an in-person farm walk at Winter Creek Farm in Kitsap County in Paulsbo. And that is scheduled for September 13th. And it's all about low till for small farms. So thank you guys. And we will connect with you again soon. Have a good night.